Okay. Now the next one is the weekend session for the January semester. The weekend section, we have some good questions. Take copy and complete the table below. So if you give that table, this list of some several organs, includes platinum, writers, and some particular. So you have to put the functions of nucleus. So what is the function? So that nucleus is the organ of blood and genetic material. It's the about the genetic material. It's what the DNA have problem. So nucleus is where the genetic information is being stored or being kept. Plasma membrane, what is the function of plasma membrane? We talk about different functions of plasma membrane. One of them is separates the intracellular and extracellular, thereby preventing or forming a kind of a barrier protecting the organelles, this cell component from the extracellular surrounding. Then that mitochondria will set the powerhouse of the cell where energy is produced. Ribosome, so basically we say this is where the protein synthesis is occurred. And then we have a polymer apparatus where we said once protein will be against the site, this is where the process packing levels and then delivered the distillation where they are needed. It's the when you have the organelles or even outside the stuff to be taken different. And the next question says differentiation between prokaryotic and eukaryotic cell. We talk about the difference between prokaryotic and cell. They are different with respect to the size, with respect to presence of nucleus, absence of nucleus. You can go back to that presence of organelles. So you can go back to that. Then the next question says define nucleic acid and state its component. We talk about nucleic acid, which is a polymer of nucleotides. Then the components, of course, we specify the component of nucleic acid, which are nucleotides. And nucleotides, we said we have different types of nucleic acid. We have DNA and RNA. There's a DNA. We have the four of them, adenine or adenosine. Triphosphate, adenosine, and monophosphate, ATP, thiamine, monophosphate, TMP, adenosine, monophosphate, AMP, thiamine, monophosphate, TMP, guanine, monophosphate, GMP, and cytidine, monophosphate, C. Likewise, in the case of RNA, you will have uridine monophosphate, UAP, without tail monophosphate, T. Then the next one says the tabular form differentiate between the yeah, we've also discussed that RNA being single stranded, RNA contain virus or the virus, and also the changes or differences in the nucleotides. Then another thing is write the full meaning of the following AMP, which I just mentioned, adenine monophosphate, guanine monophosphate, uridine monophosphate, and cyclic adenine monophosphate. Question 3 says define the following term: isomerism, aneurysm, weakness, pyronism, pyronism, housing, 3, and 4, 5. Please go back to where we define all those things. Isomerism, aneurysm. I think the only thing I will say about aneurysm is remember when we discussed about cyclization, where we have formation of a new asymmetric center, where we said either the carbox, the carbonyl carbon, or the ketone carbon in aldose and ketosis undergo cyclization. Initially, the carbonyl carbon and the ketone carbon are not asymmetric center. They are not chiral carbon, so that when the cyclization occurs, they become chiral centers. And just they become a chiral center, then they can compare isomerism. So when we have an OH group position, the top or below, it produces this, what we call anomalous. When it is up, we call it alpha anomaly. When it is down, we call it down. So the whole process is called anomalism. The process of obtaining the alpha and the beta elements is what we call anomalism. So this is also a form of isomerism that occurs as this anomeric carbon. So isomerism that occur within the anomeric carbon or due to the formation of anomeric carbon is what we call anomalism. So the next set of impulses is called the uncompleted table. Amino acids, letters, 3 letters we are given, and we are expected to put 1 letter. So we have asparagine, which is iron, arginine, which is iron. We have asparagine, which is E. We have aspartic acid, which is B. We have glutamine, which is Q. We have glutamic acid, which is E. We have tyrosine, Y, phenylalanine. In most cases, you discover that we say that the 3-liter abbreviation usually are the first 3 liters. So some of those ones that were given were not the first 3 letters, like asparagine, we have as N, and then the glutamine, which is GLM. For the 1 liter, in most cases, you say to the first liter. In all the cases here, we're given are not the first letters, and remember, I've already told you that some of them, of course, were not the first letters. For arginine, we have R. For asparagine, we have N. For acetic acid, we have D. For glutamine, we have Q. There is not even Q in the glutamine. Glutamic acid, in tyrosine, we have Y. Then in alanine, we have F, base for everything, alanine. Now the next question is say give 2 examples of each of the following amino acid with 5 chain containing hydroxyl group. We have serine, threonine, and other one that was mentioned here is tyrosine. Right? And then we have amino acid with aromatic cytosine, they have phenylalanine, they have tyrosine, they have tryptophan. We say 2, so we have the 2 here. Even the one the others containing have even the sulfur containing, we have methyl cysteine. That's playing the role of proteins in biology. That one also we don't have to get it balance. A lot of them you have them here also. And then catalysis, transportation, storage, coordinated movement, talked about actin and myosin. We talked about cytoskeletons, yes, microproline. We talked about the mechanical support, collagens, immune protection. We talked about what we call this, antibodies. Nerve imports, growth and differentiation. We all talk about this in the now question 6, which is the final one, says briefly explain the following term in the cooperative enzyme. This is our recent discussion. Let's take it what you want to think about the difficulty with the speed of the chemical reaction. Then explain how those take place or how it's the speed of a chemical reaction. The cofactors are a small non-protein molecule that associates with the active site found near the active site, the timing or health independent of the suspect or an enzyme to the particular suspect. Equal enzyme. So when we say when we have the cofactor that is non-protein but an organic substance, which is usually a derivative of protein. I would say a prosthetic group usually is tightly bonded. Then B says mention 5 general properties of enzymes. We talk about properties of enzymes, specificity, equilibrium, pH stability, temperature stability, concentration, how concentration happens, then, and then inside the classification of reasons, oxidoreductase, transferases, hypolases, lyases, isomerases, just to mention the classifications. We said there are six. So I think this is all, and this is actually what is expected from you. So please, this is how you write your notes, your discussions, and if you have any question, you make sure that you ask your question through the WhatsApp group, or you can just type it there. I'll be prepared to respond whenever I get the time. Now to your exams time, I wish you guys all the best.